I'm gonna go ahead and get started with our week 6 review. So I'm going to be reviewing basically the content that we covered for chapters 9 and 25. Remember, chapter 9 talked about basically microbial ecology. Chapter 25 dealt with like environmental side of microbiology. Chapter 9 also dealt with like nutrition and stuff like that, whereas chapter 10 was our metabolism chapter. And then we're going to jump into our most missed topics. So starting with chapter 9, we talked about the different trophs. So basically our different trophs dealing with how organisms get their carbon or their energy. And if you get your carbon from inorganic sources, so this would be like CO2, like plants do, and these are autotrophs. If you get it from organic sources, these are heterotrophs. All right. And our energy sources, your energy can come from the sun, and these are called phototrophs, or your energy can come from food chemicals. And these are called chemotrophs. All right, so a chemoheterotroph, that would be like us, we get our carbon from organic sources, and chemo because we get our energy from the organic sources that we get our carbon from by breaking them down and releasing the energy and turning it into ATP through aerobic respiration. So, excuse me. So those are our trophs, basically where we're getting our energy and our carbon from. Remember, we have talked previously about a growth factor. That's anything that has to be given in the diet for these organisms. Saprobes break down dead or decaying materials. A halophile, these guys flourish in salty habitats. Thermophiles, they grow well at high temperatures. We also talked about mesophiles. These grow at like basically medium temperatures. All right. And psychrophiles. These grow at low or cold temperatures. All right, we talk about diffusion and osmosis. Diffusion is the movement of molecules from a high concentration to a low concentration, which is driven basically by physics. And diffusion of water is called osmosis. Things that are dissolved in a liquid are called solutes. The liquid that's doing the dissolving, like water in most biology, is called a solvent. Our membranes are selectively permeable. It allows water and other small, very, very small nonpolar molecules to diffuse freely across the membrane, but most other molecules cannot cross the membrane. So a lot of our trying to balance out concentrations inside the cell versus outside the cell is dependent on that selective permeability of the membrane. So water can cross freely, but other molecules cannot. This brings us to our concepts of isotonic, hypotonic, and hypertonic. In iso, we have it equal on each side. So we would have water being able to cross freely at all times in and out of the cell. All right, and then for hypo, we would have less solute. All right, so this is whatever's dissolved in the water outside. So that would look something like this. Here's your cell, and you have all this stuff maybe inside of it, and you have less concentrated outside. So what happens is the cell tries to water itself down what's inside to match what's outside. So water would be able to go in, and then this would mean that cells would swell. In hypertonic, what you have is cell that has less in it, and what's on the outside might be very concentrated. So what happens here is this stuff would be like salt or sugar or something like that. All this very concentrated on the outside. That what the cell can do, it will try to water down the outside, or at least do so until its inside matches the concentration of the outside. And so cells will shrink. And this is a process called plasmolysis. Not phasmolysis, plasmolysis. Or plasmolysis. All right. So that's that. We can also have, if you wanted molecules to be allowed to move across the membrane of the cell, due to diffusion, we can have facilitated diffusion where we can build in these like channels basically that would allow molecules to be able to go in and out of the cell. Oh, come on. Through the channel. And if as long as it is moving by diffusion, this is facilitated diffusion. But once you are going against a gradient, that's no longer diffusion. Or if you want to go faster than what traditional diffusion would allow you to do, then now you need to be adding in energy, and that's called active transport. So moving things against a gradient or faster than what the gradient would typically allow, you need to be adding energy for that. And this includes bigger processes like phagocytosis, where we're taking up big molecules or big entire organisms by enveloping it with like the cell's membrane. All right. Right, so back to this concept that we were up here, our mesophiles, our psychrophiles, and our thermophiles. This is dealing with temperature tolerance on here. And then also dealing with oxygen. So we would have our organisms that are going to be aerobes, obligate aerobes. They need oxygen in order to survive. We have facultative anaerobes. These guys will, facultative anaerobe. These guys would prefer to use, would prefer to use oxygen if it's available. However, if oxygen is not available, they will undergo fermentation. All right, so that's a type of anaerobic mechanism to create energy. Microaerophiles can survive with less oxygen, and we have our aerotolerant anaerobes. These guys can survive in oxygen. But they don't use it. So they don't need it. Whereas obligate anaerobes, these guys cannot survive in oxygen at all, and they don't need it. Cannot O2. All right, then that brings us to our concepts of symbiosis and the things that are relating to that. In symbiosis, this is when two organisms' livelihoods are tied together. At least one of those organisms depends on that relationship for it to survive. So there's three major groups that will fall into symbiosis. This is going to be commensalism. In commensalism, one of the organism's benefits, it's called the commensal organism, and then the other one is unaffected. And we have the mutualism. Both organisms are benefiting. So they're either like, for example, the bacteria in our gut are getting shelter and they're provided with nutrients from our food in our gut. Whereas we are getting our food broken down by them, and then we can absorb those nutrients more easily. Plus, we have the protection called microbial antagonism, which we learn about in chapter 13, about how microbes can actually provide us with protection from pathogens, as long as we are maintaining our population the way that it should be, right? So that's a mutualistic relationship. We're getting benefit from them and they're getting benefit from us. And then we have parasitism, where one organism is like basically feeding off of the other organism, causing it harm. So if one organism is harmed in that relationship, that's parasitism. And that includes things as obvious as like ticks, which have to feed on animals, right? That's their livelihood is tied into that. And the animal is always being harmed by that. This would also include things like viruses, pathogenic bacteria, and like our helminths and things like that, causing us harm while they're feeding off of us. If an organism is an obligate parasites cannot survive outside of their host. And so they, that means that it doesn't have the option to be parasitic or not. Now, let me explain that. So obviously we've talked about ticks and how ticks have to feed on animals. Those are obligate parasites. We talk about something like the tuberculosis bacterium. It doesn't ever exist in a sort of any sort of benefit to the host, and it's always causing some kind of harm to the host, right? But when we talk about E. coli in your gut serves you good, but it does gut bacteria stuff. But if it moves out of the gut to somewhere else, like into your urinary tract, then it shouldn't be there and it can cause harm. So it's not an obligate parasite, right? It can form a parasitic relationship with you if it's living in your bladder, but normally it doesn't do that. So it is not an obligate. It is not forced to exist that way. We talk about biofilms and how bacteria can communicate with each other using quorum sensing, basically responding to chemicals made by the nearby bacteria to determine the health of that community and whether or not they want to stay in that. Then we had our growth curves. This was our lag phase, our exponential or our log phase, our stationary phase, that's supposed to be a flat line. And then our death phase. So the lag phase is basically where we are just adjusting to our environment. Then the log or exponential phase, I'll put expo for exponential. This is a period of literal exponential growth. We're just doubling, and then that's doubling, and then that's doubling, and then that's doubling, and so on and so forth, because everything's good. We have all the resources that we need, and we can reproduce. Stationary phase. This is where growth has slowed down. We're not multiplying as much. Maybe the resources have allowed us to reach a sort of equilibrium of growth versus death. But then once death starts to be the dominant thing, we're running out of resources. There's too many toxic waste products and things like that. That's when we start declining in our population, and that's the death phase. All right, moving on to chapter 25 then, talking about the carbon cycle. You would have CO2 in the air, and then there would be plants or something like that would be on the ground. Those plants are going to convert CO2 into organic molecules like glucose. That organic carbon will then go into, I don't know, cows or something like that. That might be, I can't draw a cow. This is me drawing a cow, okay? But anyways, this is our happy little cow. He's got lots of spots on him. This thing won't let me draw spots, so it is what it is. But he's happy. He's going to eat those plants. So he'll get those organic molecules so that he can exhale carbon dioxide as waste. That is that simple. We also have things that are going to eat the dead cow and then get the carbon dioxide from, and then get the organic material from the cow. And then those guys will also release carbon dioxide. We also have normal natural processes in just the planet, like combustion of volcanoes and things like that, burning fires, that'll release carbon dioxide as well. But those all play a part in that carbon cycle. Methane is also important, where this cow might have bacteria living in its booty, well, not in its booty necessarily, but in its guts. and then release from its booty the methane. And methane is CH4, and it's actually a more potent greenhouse gas than CO2, and that these are going to increase, basically, create global warming by creating a sort of blanket of these carbon compounds in the atmosphere that allow heat to get bounced off of that atmosphere instead of being able to be released out into space, or becoming to part of higher parts of the atmosphere, creating that heat basically being held in, creating a warmer planet and leading to global warming. Next, we have nitrogen. I don't need you guys to necessarily know the difference between ammonification, nitrification, or denitrification, but I'd be aware that these are names of processes that are involved in the nitrogen cycle. Plants cannot get nitrogen from the soil straight up. It has to be processed by bacteria. Bacteria process it into a form that can then be used by the plants. So these processes that are involved in that are ammonification, where we'll produce ammonia. We have nitrification, where we would take ammonia and changing it to nitrite, and then turn that into nitrate by other bacteria. And then denitrification, taking nitrate and turning it into atmospheric nitrogen. All these are going to play a role in the basically the cycle of nitrogen in our environment. Again, you just need to be able to recognize that ammonification, nitrification, and denitrification are processes involved in the nitrogen cycle. Okay, that's all you need to know about them. We talk about marine microbes and the fact that plastic that has created reefs in our oceans, and microplastics that are affecting our environment, basically affecting the entire ecosystem of the ocean and aquatic environments, because it's creating basically a film on the surface of the ocean that's preventing the growth of microbes near the surface. And then everything that feeds off of them is in effect affected by that. So it's creating a very massive problem. About 88% of the ocean surfaces is covered in microplastics or plastics. So it's pretty disgusting. We have water filtration and wastewater treatment. I would be aware of the fact that these processes do occur in several steps, and that at each step has its purpose, right? Well, if we're talking about sedimentation, sedimentation is exactly what it sounds like, allowing things to settle. So allowing solid materials to settle, sedimentation, and then filtering, again, what it sounds like. You can filter it through a carbon, like activated charcoal and charcoal filters and things like that, or just literal like actual filters that are made up of mesh, and different steps of that. And then we have treatment with like chlorine or chemicals. So chlorine or UV radiation in order to kill off any microbes, and then it can be treated. Wastewater treatment, which is talking about actual like feces or what we would consider to be what we call like brown water or black water, coming from toilets and things like that, has to be treated just in a little bit more strict levels of these steps. Other than sedimentation, we also have a sludge phase. And that sludge phase, in addition to bioremediation, so we introduce bacteria and fungi and things that break down the material like fecal matter and things like that into something that can then be separated out a little bit more easily before we go through with our filtration and everything, and then releasing that water back into the environment. We have to be able to monitor all of those steps as well and make sure that the water that we are drinking or the water that we are releasing into the environment doesn't have too many contaminants. So one of the ways that we do that is we check for something called fecal coliforms. And coliforms are indicator bacteria. They themselves might not be pathogenic, at least not to otherwise healthy people. But if they got into your water sample, then the pathogens could get there. So they indicate to us that pathogens could get through our filtering process. And so we monitor that pretty regularly in order to make sure that the water that we are releasing back into the environment, or that we're drinking or washing with or whatever, doesn't have the potential to have pathogens in it. Water that is free of pathogens and is free of obvious defects, looks clear, doesn't smell crazy, and is free of pathogens. We call that potable water. And so access to potable water is incredibly important for the health of the society, right? So That's important to remember as well. We know we use microbes in our food, whether we're talking about making bread, beer, wine, kimchi, sauerkraut, your yogurt, cheese, all of these things involve using microbes. Some of these use bacteria, some of these use yeasts, which are fungi. No matter what, a lot of these guys depend on fermentation, which we'll talk about in chapter 10, as one of the processes that organisms will use in moments where aerobic respiration isn't possible. So lack of oxygen. So organisms that can switch between fermentation and aerobic respiration, those guys are called facultative anaerobes, right? Which we talked about just a second ago. Then we have the effect of climate on disease and the idea of One Health. And so one thing that's really important is to remind everybody that as the planet warms and temperatures get higher and higher in areas where it was previously lower, and if you've lived in Oklahoma long enough to be an adult, then you probably remember when you were a little kid and we used to have pretty heavy snows in Oklahoma pretty reliably. We used to have white Christmases and all of this stuff. I used to build igloos in my front yard when I was a kid. But now the likelihood of snow being that reliable is pretty low. I mean, you're lucky to get as much snow as we did this last one snow session. And it used to be snowy pretty much most of the winter. And that's an effect of global warming. And the warmer that it gets, the further north, the more likely it is for mosquitoes to be able to migrate up north and live longer, and therefore also possess and pass on the diseases that they can potentially carry with them. Things like malaria, dengue fever, and certain hemorrhagic fevers that are associated with, mosquito transmission. It's why we saw such an uptick for a little while with West Nile virus. Don't think for a minute that West Nile virus isn't around. It's just that most people that contract West Nile virus don't have symptoms. Most don't. Some people will develop encephalitis that can potentially be deadly as a result of it, but we wouldn't usually see that in cooler climates, so the further north that temperatures are getting warmer and warmer, the more we're going to see spread of diseases like that. And things that we've seen further south of us, like dengue, are becoming endemic here in the United States now. And don't think for a minute that's any small thing. It's a pretty horrible disease to have. Dengue fever is. All right, other effects of climate change, talking about like urbanization, so moving into wild habitats, and that's going to expose you to diseases that humans normally wouldn't be exposed to. And let alone migration of mosquito populations further north and affecting populations that normally don't see those diseases. Also talking about drying out tropical areas near the equator that now might become more desert-like and can't support the life of the bacteria that normally would convert nitrogen into a usable form for plants. And that can lead to, it already has started to lead to famine in certain areas of the world. Chapter 10, we talked about metabolism. Metabolism is all of the chemical processes in the cell and in an organism. And it can be broken up in basically two sides. Catabolism, which is breaking, so catabolism is basically breaking things apart and then getting energy from breaking it apart. And then anabolism is building new molecules, and that usually requires energy. And a lot of these chemical reactions in the cell are basically governed by enzymes. And enzymes, again, we've learned that enzymes are primarily proteins. These proteins are catalysts, so they're going to lower activation energy. So just relying on amino acids to naturally bond together to create this protein that you want, or to drive these chemical reactions that we usually have in the cell, that enzymes are helping those things happen. So they're not just relying on spontaneity, but allowing us to have control over them. So it's really what these enzymes are allowing us to do. The thing that the enzyme binds onto and acts on is called the substrate. Some enzymes consist of two parts, essentially. So you will have the protein part. This part is called the apoenzyme. And then you'll have the non-protein part. This part is always called the cofactor. If this is inorganic, including metals, like metallic ions, like what we have with iron in your hemoglobin, for example, that this is still a cofactor. Okay. But if it is organic, I should have written this differently, but yeah, if our cofactors are organic, it's called something else. It's called a coenzyme. Right? And that this whole thing, protein plus non-protein, this whole thing is called a holoenzyme. And it sounds like whole to me, like you're reading the whole thing. Holoenzyme, even though it's just not with a W, right? So that's how I remember what those are. All right, there are different classes of enzymes based on what they do. The 6 major classes of enzymes that operate within cells are the oxidoreductases, which are obviously involved in redox reactions, which I'll go over in just a second. Well, I might as well go over it now. A redox reaction refers to reduction and oxidation. And this is basically the transfer of electrons. So if you're transferring electrons, if you're helping in the transfer of electrons, this is an oxidoreductase. These are enzymes that help in the transfer of electrons. If you gain an electron, then that is reduced. If you lose an electron, that is oxidized. Okay. So that's basically what oxidoreductases are going to be doing. They're going to be involved in the exchange of electrons. Then we have transferases. Transferases basically moving whole groups off of one molecule onto another molecule. This could include transferring phosphate groups or something like that. Then we have the hydrolases that are involved in hydrolysis. This is basically breaking apart water in order to break apart other molecules. So it allows you to break apart larger molecules into smaller parts by breaking apart water. And then we have the ligases, which is joining things together, and that creates water as a product. It's basically the opposite of the hydrolases. So hydrolases and ligases are pretty much the same thing. Ligases, these deal with double bonds. And isomers basically are dealing with the change in shape of molecules. In order for proteins to do their jobs properly, their shapes are incredibly important in how they do their functions. And their shape is dictated basically by the R groups on the amino acids that make up the protein. So how those amino acids are linked together, and then how those will basically react with each other, next to each other, or across from each other. And so that its main structure and its ability to function because of that structure and chemical makeup is the natural form. If you have a denatured enzyme, then now that is protein that is misfolded. You can cause proteins to misfold for a number of reasons, but pH, heat, and chemicals are the main ones. We'll be talking about how exactly those affect proteins and how we can use that to keep things sterile or at least sanitary for whatever reason when we get into chapter 11, when we get into unit 3. But the denaturation of proteins depends on breaking those weak hydrogen bonds, which are dependent on attractions between or repulsions from charged or uncharged R groups on those amino acids. And so we can disrupt that by having too many protons or too few protons, or just by adding some heat, like we talked about with denaturing DNA. It works the same way with proteins. Then we can also control our protein function, not just by denaturation of the protein, but by including inhibitors. So proteins can be inhibited in 2 major ways. We have competitive inhibition or non-competitive. Competitive inhibition means that the inhibitor, whatever it is, binds at the active site. So competitive inhibition, you have an inhibitor literally binding in the active site where the substrate normally sits. So we're normally acting on our substrate there at that site. This guy comes in and binds there and makes it so that the normal substrate can't bind there. So Non-competitive inhibition means that your inhibitor is binding somewhere else, not the active site. And so the place this somewhere else is a site on that enzyme called the allosteric or regulatory site. All right, so we already talked about redox reactions, so I'm going to go back over that one. And we know ATP is our energy molecule. It's adenosine triphosphate. So adenosine, what that means is we have that nitrogenous base, adenine. It's the same as what makes things A, T, C, or G. This is the A part of the A, T, C, or G in like DNA or RNA. So adenine, and it's linked onto a 5-carbon sugar. This happens to be ribose, like it's in our RNA, not deoxyribose. And that this is linked on to 3 phosphates. So this all right here, this part right here is a nucleotide. This nucleotide is what would be making up like our RNA. And I'll just be repeating units of that, A, T, A, U, C, G, whatever it is for our RNA. But you take that nucleotide with that A on it and tack on some more phosphates. Those phosphates are all negatively charged. And so the fact that they are negatively charged, they're all repelling each other. And they don't want to be next to each other, even though they're covalently bonded. So they're sharing electrons. All they want to do is get away from each other. And so whenever you take off one of those phosphates, each time you take off a phosphate, it releases energy and the cell can use that to power basically its processes. The whole point of aerobic respiration is to generate ATP. So let's get into aerobic respiration really quickly. I don't have time to redo, everything we need to know about it. Let's give a basic summation of aerobic respiration. Aerobic respiration depends very highly on electron movement. And so the whole point of it is to collect as many electrons as we can and then shuttle it through our electron transport chain. And that by shuttling the electrons through the electron transport chain, which is a set of proteins called cytochromes, that each of those cytochromes gets, basically changed by the electron that's moving through it or interacting with it. And that causes protons to be pumped outside of a membrane. Those protons then build up in a gradient outside of the membrane, and they want to come back in because diffusion, right? It wants to equal out on both sides. The only way it can come in is through ATP synthase. And then ATP synthase, when the proton moves through it to come back into the cell where it's just trying to equalize on both sides, when it does that, it allows ADP, adenosine diphosphate, to gain a phosphate onto it, and then that creates ATP. And that's how ATP is made. How does all of this happen? Well, the first step is glycolysis. In glycolysis, you're basically taking glucose, which is 6 carbons, and turning it into 2 3-carbon pyruvates or pyruvic acid. All right, so that's the first step. And there's an intermediate step where we are going to be taking that pyruvate, each of those pyruvate, and turning it into acetyl-CoA. As it turns out, in glycolysis, you will be making about 2 net ATP. You'll be making 4 altogether, but you'll have 2 of them get used up just because that's how glycolysis works. You use ATP and you make ATP. So net, you'll have 2 ATP made. Then you'll have some NADH made, which is carrying an electron. This is the important part of it. But you'll make a little bit of ATP, you'll make a little bit of NADH, and then you'll make this pyruvate, which the pyruvates are the big daddy take-home message of glycolysis. All right. Then this intermediate step turns it into acetyl-CoA. From this, we do get some products as well. We're going to get a little bit of CO2 and some NADH here. This is the first time we're going to release CO2 as a waste product. Then in the Krebs cycle, we're going to basically cycle that acetyl-CoA through and collect electrons in the form of our NADH and FADH2. Okay, so we're stealing that off. We're also going to make a little bit of ATP and a little bit of carbon dioxide as well, right? And so that's a major step. So we have three major steps: the glycolysis, the Krebs, and then the electron transport chain. Where, like I said earlier, you're basically going to have in the electron transport chain, I'm going to draw it up here in this corner up here. Up right. So the electron transport chain, the way that it works is you have your cytochromes that are sitting next to each other in the membrane. And these are just proteins that are sitting in the membrane. Here's the membrane. All right, so here's our little lipid bilayer, whatever. That's our membrane there. All right, so basically what we're going to do is take NADH and turn it into, it's going to give off waste of NAD that can be reused again. So we don't need that, but then we do need the H, and we need the electrons that come with it. So the H will give it up its electron, so it's separate now. It'll be H pluses, and it'll be electrons. The electron goes through all of these cytochromes here. As it travels through there, the protons will get pumped out outside. So you get all these protons, positive charge out here, as that negative charge electron moves through those cytochromes there. Then further downstream, you have ATP synthase, which is an enzyme that these pluses can then come back in to try to equal out the gradient. And that when that happens, a DP will become a TP. And this is the big goal for that. You also have this buildup of H pluses, and you'll have this buildup of electrons that are going to come through here. And so these electrons and these H pluses and some O2 will all come together. These three here come together, and then that'll create water as a waste. This is why oxygen is the final electron acceptor, because as these electrons are moving through this, the electron transport chain here, up here, these electrons have to go somewhere, and so they're going to get accepted by oxygen. Oxygen's going to gladly bind with them, and it's going to get stabilized by the addition of these protons, which is going to help eliminate those guys so we can keep building up that gradient. Because as soon as you get rid of oxygen, you can't get rid of those electrons, you can't get rid of those protons, we can't maintain that gradient. And so we will just have the same amount of H pluses on each side. They no longer keep moving through the ATP synthase at the rate that we need it to in order to make enough ATP to power your body, which is why you suffocate and die. So that's essentially aerobic respiration in a nutshell. Bacteria and archaea, these guys can be aerobic respiration guys, but this won't occur in the mitochondria like it does for us. For eukaryotes, aerobic respiration occurs on the inner membrane of the mitochondria. For bacteria and archaea, they do not have mitochondria. So what they do is their aerobic respiration occurs on their inner or cytoplasmic membrane. So all of this mess that happens up here for electron transport chain, that happens on their actual cytoplasmic membrane. So it still happens on a membrane. They're just going to be pumping those protons outside of that membrane into the space between their cell wall and their inner membrane. Okay. We will make anywhere from 36 to 38 ATP. If you have mitochondria, you'll make 36. If you don't have mitochondria, if you're bacteria and you're just doing this on your cytoplasmic membrane, you'll make 38. The reason that you have 2 less ATP if you have mitochondria is because you have to move products in and out of the mitochondria, and that costs you some energy when you do that. All right, amphibolism is a term that we can link that energy production for catabolism, which is what aerobic respiration is. Aerobic respiration is a catabolic process. We are breaking apart glucose and then turning it into ATP energy. So that is catabolism, and is the best example we have of catabolism. So if you want to link that creation of ATP or any of those intermediates in that process to building other bigger molecules, then that's the anabolic side of it. So linking the catabolic to the anabolic, that is amphibolism. All right, fermentation, if you do not have oxygen, the only thing you can go through is glycolysis. You'll make a little bit of ATP, but you need to get rid of these electrons that you made on this NADH right there from glycolysis. So the way that they do that is they just feed it back into their pyruvate waste. For them, pyruvate is a waste molecule. And so they'll feed their electrons back into that. And that's where we get the production of things like alcohol, lactic acid, and carbon dioxide from fermentation. And they'll just keep pumping through glycolysis. They don't have the ability to go past that point if they have to ferment. In photosynthesis, there are two steps involved in photosynthesis, the light cycles and the light independent cycles. In the light cycles, they basically use light. This should look familiar because it's basically the same thing as what's above, up there. So light excites these guys, which are our pigment molecules for them. And they work a lot like our cytochromes, okay? So light comes and excites those guys. Then we have our H pluses that can create this gradient out here. Again, the proton motive force is what this is called. And then those guys can come through ATP synthase the exact same way, and then we'll get ATP made. Make a little bit of ATP just by getting some pigments excited on a membrane. And that's entirely what the light cycles of photosynthesis are for, just to make ATP. The light independent reactions, though, they take that ATP and essentially take the ATP. It's a big cycle with a whole bunch of molecules that are involved, and the cycle is called the Calvin cycle. And in the Calvin cycle, they're going to use that ATP from the light reactions, and they're going to take carbon dioxide from the atmosphere, and with a bunch of intermediate molecules and some very complicated stuff, they're going to make glucose. What are they going to do with that glucose? Well, they can use that to build structures of the actual cell. So that's where they get their organic molecules from. Not from eating it, not from absorbing them from their environment, from creating them from carbon dioxide. And so they create all of their organic molecules from scratch from carbon dioxide. And glucose is one of the intermediates there. But they also will use glucose made from the ATP from the sunlight and the carbon dioxide from the air, ATP from sun, carbon dioxide from air, right? To make the glucose here that they will just feed right back into aerobic respiration. So things like plants, they also, or any photosynthetic organism, typically also need oxygen in order to drive the aerobic respiration side of things in order to get enough energy to sustain their existence. Plants have mitochondria too. And what happens in mitochondria? Aerobic respiration. So that's what photosynthesis is all about. All right, so let's go over our most missed topics for these chapters. Back to chapter 9. Osmosis and the tonicities. Remember, osmosis is the movement of water in the diffusion of water across the membrane in order to, in order to keep things equal. So people are messaging me and it's just distracting me. I have very limited ability to pay attention to multiple things at once. So osmosis, water moving across the membrane, trying to keep the concentration of water the same on each side. So when that concentration of water is disrupted by things like salt being added to it or sugar being added to it or something, we want to keep the concentration of water balanced on each side. And it can cross the membrane. The salt or the sugar can't cross the membrane on its own, right? So that leads to tonicity. So tonicity, things that are dissolved in the water and trying to keep the concentration of those things and the water balanced on both sides. When they are balanced, that is called isotonic. When there is less solute, so less salt or sugar outside of the cell than what is inside of the cell, that's a hypotonic solution. And what happens is because the cell can't give its salt or sugar out of the cell to basically make things equal on both sides, it can move only the water. And so it tries to water its insides down in order to match it to what's outside. And so cells will swell up. And hypertonic, this is, I wrote it on the wrong thing, didn't I? Right. So that's hypotonic. It's my fault. I don't know what I was writing on. So in isotonic solutions, the water moves, it's equal on both sides. But water still moves, it's just equal the whole time. In hypotonic, in order to equal out the two sides, the cell will basically take up water and swell. It can burst even. In hypertonic, the cell will basically push water out to try to water down its outside or increase the concentration on the inside, both simultaneously, so that its inside salts and sugar will equal the concentration of the salt and sugar outside. Eventually, that'll happen. But when it pushes this water out, cell shrinks. We know that. If you put something in salt, it's going to draw all the moisture out of the cells. So the cell will shrink, and this is a process called plasmolysis. All right, so our little mnemonic here was I hope hypercells, or IHHC. So isotonic is the I, no net movement of water in and out is the same, it's equilibrium there. Hypotonic, water moves into the cell. Hypertonic, water moves out of the cell. And all of this is dependent on that selectively permeable membrane of cells. So here's our questions relating to this topic. Which of the following best describes the movement of water in an isotonic solution? Now, we need to remember that if a solution is isotonic, this is our equilibrium state. That does not mean that water doesn't move. It's just moving in and out at equal rates. We're not moving in and swelling. We're not moving out and shrinking. It's just both at the same time equally, okay? So what are our choices? Water moves into the cell, causing it to swell. No, because we're at equilibrium, we shouldn't be swelling or shrinking. So our next choice is water moves out of the cell, causing it to shrink. Again, we're maintaining equilibrium. We shouldn't be swelling or shrinking. Water moves in and out of the cell at equal rates. This is exactly what happens. It's going to be C, because it's never actually 100% perfect. The closest we can get to it is isotonic. And because chemistry, don't we love it? And physics, oh my gosh, not confusing at all. That these will dictate that the water molecules are always moving. So they're just going in and out at equal rates. And then we have water does not move across the membrane in an isotonic solution. That's incorrect because it is moving always, even when it's at equilibrium. It doesn't just sit there. All right, our question is now a patient is receiving intravenous fluids and the physician orders saline solution. It's 0.9% saline. So when you administer a patient saline, what type of tonicity is that saline solution and why do we choose it? So you would hope that the reason that you would be giving your patient this saline solution is to give them a solution that is supposed to be best suited to keep cells hydrated, right? So you don't want something that's hypotonic. You don't want water to enter into the cells because you could be causing damage to them by causing them to swell. You don't want something that's hypertonic because you don't want to draw the water out of the cells and dehydrate that patient even further. Their cells need the water in there. You want something that's going to be isotonic. We have two choices with isotonic. Don't need to jump ahead of myself. You want something that's isotonic for sure. So what's the differences between our choices here? Does it maintain the balance of fluids between the inside and outside, or does it cause the cells to shrink? Obviously, we're maintaining that. We're not causing any shrinkage of our cells. We want this nice happy equilibrium so we can maintain the fluid balance that's supposed to be in the cells as best as we can. So isotonic compared to what we would expect a healthy cell situation to be. Moving on to chapter 25. This is talking about the effects of global warming, which is bringing us into that mindset that as a temperature increases in our climate, the mosquitoes can live further north, and the diseases that they can carry will also be coming with them. So this is essentially talking a little bit about the effects this can have in a little more detail than what we talked about earlier. Hot temps allow disease populations. So obviously hot, dealing with higher temperatures, hot temps, higher temperatures, allowing for disease populations. So disease carrying populations of mosquitoes will increase as the higher temperatures rise further north. So how does global warming contribute to the increased risk of mosquito-borne diseases? Global warming means that the further north that we go, the warmer it'll be, and then the mosquitoes will be able to survive better, right? So we're having increased mosquito population and the diseases that they carry. All right, so what are our choices? Does it decrease the breeding sites for mosquitoes due to less rainfall? Well, no. We're talking about an increased risk of mosquito-borne diseases. So if you were decreasing the breeding sites, you'd have decreasing mosquitoes. That's not what we're talking about. Is it reducing mosquito population? It's not reducing, it's increasing. It literally tells us we're gonna have an increase in mosquito-borne diseases, and that's gonna come with an increase in mosquito populations. So we know we're expanding the mosquito habitats and accelerating the development of the mosquitoes themselves, as well as the pathogens that they carry. And then D, it prevents replication of pathogens within mosquitoes, don't we wish? That's why C is our correct answer. Our next question, which of the following public health strategies is most likely to be affected by the expansion of mosquito habitats due to global warming? Okay, so reducing antibiotic prescriptions to prevent antibiotic resistance, is that going to be affected by our mosquito populations? This doesn't have to do with our antibiotic resistance. That happens as a result of our bacteria being able to exchange the genetic information between each other through horizontal gene transfer, which we learned about in earlier chapters. So that was chapter 6. That doesn't rely on mosquitoes, right? So next choice, enhancing vaccination programs for mosquito-borne diseases in newly affected regions. You would want to be doing that. You want to protect the populations further north from things like dengue or yellow fever or other things that are transmitted by mosquitoes if you have vaccinations available. And if you don't, if there's more population that you might want to protect with that vaccine, you might want to develop a vaccine for those. Promoting low-fat diets. Well, you might want to have a decrease in obesity-related diseases. This is not relating to the expansion of the mosquito habitat. Increasing the use of personal protective equipment in non-endemic areas. Personal protective equipment isn't going to save the populations from the mosquito diseases. Now, you might consider then using, personal protective equipment, going out into environments where there are mosquitoes, but this isn't going to affect that healthcare environment and dealing with that. So B is our correct answer there. Moving on to chapter 10's most missed topics. All right, the parts of the enzyme. So the whole enzyme is the holo enzyme. The apo enzyme is the protein component. The cofactor is the non-protein. These two things combined make up the holo enzyme, all right? The area where our molecule that's going to get acted upon, which is called our substrate binds at the active site. All right, our cofactors can be things that are inorganic, like metal ions. These will be important for the structure and the function of the enzyme, and it won't be able to act without them. They can even actively participate in the process. You could have organic molecules, like these are coenzymes. In fact, NAD and FAD are also coenzymes that are going to be carrying our electrons for us. And so they actually are going to help with our oxidoreductases in transferring those electrons around. Then we have the substrate itself. Again, this is going to bind in the active site. And then our little mnemonic here was here, all apes can stay. So here is holoenzyme, all is apoenzyme. Apes is active site, or I guess you could switch those either way, but your active site is going to be present on the apoenzyme. FYI, it's always present the active site is always present on the protein portion. So the cofactor does not bind at the active site itself. It could be active near the active site, but it is not the active site. It's not binding in the active site, but it can participate near the active site. And we have CAN, C is about the cofactors. Again, this is the non-protein portion that is needed for conjugated enzymes. Simple enzymes don't have cofactors. If your cofactor is an organic molecule, like vitamins and NAD and FAD, for example, it's organic, then it is a coenzyme, is what we call it. All right, and the substrate is the stay part of the mnemonic. So our questions for this one, for our enzymes: which of the following enzyme components is directly responsible for the chemical reaction by binding to the substrate? Well, we know that an enzyme is made up of an apoenzyme, which is the protein part. And we know that it has an active site where our substrate binds, which is the molecule that we're acting on. And then we know that this is part of the apoenzyme. And the other part of conjugated enzymes is called the cofactor. The cofactor is going to be a coenzyme if it's organic molecule. Literally carbon-hydrogen backbone, right? And these together form the holoenzyme. All right. So I have all my little notes down there to answer these questions now. So which of the following enzyme components is responsible for the chemical reaction by binding to the substrate? Where do we have binding of our substrate? That's the active site. So we're able to select that out of here. Remember our cofactor and our coenzyme. Our coenzyme is a cofactor. It's just organic version. These guys aren't binding to the substrate, but they are essential for the holoenzyme to function. The apoenzyme is just the protein portion. All right, next question. In the context of enzyme deficiencies leading to metabolic disorders, I have a lot of these types of questions on this review session. So we have a metabolic disorder where we have an absence or dysfunction that affects enzyme activity and could result in conditions like phenylketonuria. Okay. So which part of an enzyme would be affected in order for us to have it so that our enzyme would no longer work? Would it be the coenzyme or the cofactor? Remember, these are the same. It's just this is just an organic version. Now, if you have a genetic disorder, your vitamins, your iron, things like that aren't what's going to be getting changed by your genetics, right? Remember, your genetics code for proteins. So we're not probably changing your cofactors there with your genetic abnormality. We're probably changing the protein. So we're also not changing the substrate, which is the thing that your enzyme acts on. So it's not going to be any of this. The protein portion of your enzyme is the apoenzyme. So this is the part that will be affected by your genetic defects, leading to your improperly working protein. All right, our next concept is redox reactions. Remember, Leo says grr. Leo the lion says grr, or you can say oil rig, where lose electrons oxidized or gain electrons reduced. And again, oxidation is loss, reduction is gain. Either way. So that's how I remember those things, and that the transfer of these electrons are really what's going to be driving our electron transport chain whenever we are creating our ATP and our aerobic respiration. All right, so I'm going to move on with the questions for this one. In a redox reaction, and remember, I'm going to put oil rig over here so that I don't forget. because I am not even kidding when I say that when I would have to answer questions about this, even in graduate school, I would have to write down oil rig so I can always remember which one was which. All right, so in a redox reaction, which term describes the process where a molecule gains electrons? So we're talking about gain, we'll find our G, that's reduction. That's B. Remember, oxidation is loss. Hydrolysis, this is talking about breaking bonds by adding water. We're going to add water. When we add water, it gets broken down too. So we're breaking water apart too. Breaking bonds by breaking water also. And then dehydration is just the removal of water from a situation. So those are not the answers. So our next question, in the management of certain diseases, such as cystic fibrosis, medications that impact oxidative stress are used. What? So oxidative. That's the O, right? What type of reaction is directly targeted by antioxidants used in these treatments? Are we targeting hydrolysis? That was this situation over here. That shouldn't be it. Are we targeting redox reactions? Here we are talking about oxidation and reduction. Obviously, we're leading with antioxidants. We're trying to prevent oxidation. So this is dealing with our redox reactions. It's not phosphorylation reactions. This doesn't have to deal with oxidation or reduction. It's not condensation reactions. This is going to be dealing with forming larger molecules by combining smaller molecules. It's the opposite, basically, of hydrolysis. Condensation reactions often have to do with ligases. Okay, remember ligases and hydrolases, kind of the opposites of one another. So no, it's not these. In oxidation and reduction, we're always talking about transfer of electrons, and so that's always going to be redox reactions. All right, our next topic, where does electron transport chain occur in eukaryotes versus bacteria and archaea? In our cells, we are so used to talking about our mitochondria being our energy source that people often get confused here because they just assume that all organisms are using mitochondria to create their energy. Whereas, matter of fact, bacteria lack mitochondria. So let's not forget that, okay? Bacteria do not have mitochondria. So while our electron transport chain, now I'll draw that out one more time to remind us of how it works. We have our cytochromes lined up to each other, and we're going to put our electrons and shuttle them through here, which is going to cause our H pluses to move out so that we can put that back through ATP synthase. Create our gradient here. Remember, our gradient is called the proton motive force. This is really not my best artwork, but that these protons are going to move back in and allow us to change a dp into a tp. So just drawing this all out so that we can visualize it one more time. This all happens on a membrane. But where is that membrane? In eukaryotes, which is like us, multicellular organisms, as well as our protozoa and our fungi, like our yeast. This is going to happen in the inner mitochondrial membrane. So your mitochondria looks like this, remember? And then it has all these like wiggly wobbly guys that this isn't nailing to, it's not letting me draw it because it's too long of a line. Yeah, it won't even let me draw that. Anyways, that this is the inner membrane. That wiggly wobbly guy. Okay, and that inner membrane is where all of this down here is happening. In bacteria, bacteria just look like this is their inner membrane and this is their wall around it. So this is thick, right? This is their peptidoglycan wall on the outside. All of this happens on their inner membrane, their cytoplasmic membrane or their plasma membrane. Same thing, plasma membrane or cytoplasmic. Right. So it says, please make it more efficient. Please make plasma membrane. This is going to be in bacteria versus mitochondrial. It's a mitochondrial like membrane. And then so that's where that's the environment. That's the E, I guess, but that's the environment where that's going to be happening. Okay. Also depends on whether your organism is aerobic or anaerobic and whether you're using oxygen or something else. All right, so where does the electron transport chain operate in bacterial cells? So remember bacteria, they've got their membrane and they've got their wall on the outside, whatever. This is all happening on that cytoplasmic or inner membrane. So is it happening on the inner mitochondrial membrane? No, because they don't have mitochondria. The outer mitochondrial membrane, they don't have mitochondria. Is it happening on the cytoplasmic membrane or the nuclear membrane? Cytoplasmic, they don't have a nucleus either. All you really need to know is that they don't have any other internal organelles. Okay, in the treatment of mitochondrial disorders, such as Leber's hereditary optic neuropathy, which apparently is a mitochondrial disorder, which part of the cell is directly targeted by therapies aimed at improving cellular respiration? Well, we know cellular respiration, also known as aerobic respiration, all happens in the mitochondria on the inner mitochondrial membrane. So if you're treating something that's involved in cellular respiration or aerobic respiration, you're going to be affecting the mitochondrial membrane, not the cytoplasmic membrane, not for people. This is a hereditary thing with optic neuropathy. It's for multicellular organisms. This is not going to be bacteria, right? And the bacteria are the ones that have this happen on their cytoplasmic plasma membrane, right? Endoplasmic reticulum, remember, this is the site for like macromolecule production, especially our protein production. This is our product line, and this is our packaging line, is our Golgi. All right, next topic, ATP generation through aerobic respiration. It's generally made by 3 major steps. We have glycolysis, which we said was glucose to pyruvate. We had our intermediate step where we're turning pyruvate into acetyl-CoA. Then we have the Krebs cycle, where we're basically stealing electrons from everything. And then we have the electron transport chain. The whole point of breaking down glucose is to make pyruvate, to turn it into electrons that we can feed into the electron transport chain. The electron transport chain is the largest producer of ATP of all of the processes in the cell by far. And this happens through a process called chemiosmosis, using the movement of, on our membrane, right? We had our cytochromes. We're moving electrons through that and then pumping H pluses out. This is all due to chemical reactions and these molecules interacting with one another, that this process and then allowing us to make ATP, right? Whenever we are moving our pluses back in, that this process is called chemiosmosis, chemical movement, chemical interactions, basically driving our production of our ATP. So that allows us to create our energy. So what is the primary role of the proton motive force in cells? So again, really quick drawing this out. That I made it put a whole ton of them there, but that's just fine. We have our ATP synthase. And then we have our pluses. These are all H pluses, by the way. And that these pluses will move back in. That when they do, it allows us to take a ADP and make it into ATP, which is the whole point of everything in here. And then this is all driven by moving electrons through here and moving H's across as we do that. All right, so that's how all this is going to work. So what is the primary role of the proton motive force? That's this. This gradient of H plus is outside. Why do we have that? Is it to synthesize proteins from amino acids? It's not the primary role of that. The whole primary role of creating that proton gradient is to drive the synthesis of ATP through ATP synthase, this channel molecule enzyme here. It's an enzyme that is also a channel, and as things move through it, causes it to allow it to add a phosphate onto ADP. So the proton mode of force, does it facilitate transport of electrons through the electron transport chain? It's the opposite. The transport of electrons through the electron transport chain drives forcing the H pluses out and creating the proton motive force. And then the proton motive force drives the synthesis of ATP through ATP synthase. And then is the point of it to assist replication of DNA? No. So that leaves us with our better choice of B. So in the treatment of mitochondrial diseases, such as mitochondrial myopathy, which cellular process is directly impacted by therapies aimed at enhancing the proton motive force? If we're trying to make more of that proton motive force, what would the outcome be? Because of that, would it affect DNA replication, protein synthesis, ATP production, or lipid metabolism? That proton motive force is key in creating ATP via moving those protons back through ATP synthase. All right, then we have our final electron acceptor in aerobic respiration is oxygen. Again, whenever we have our cytochromes and we're moving our molecules through all this, we have our electron getting moved through here, we have our H plus getting moved out, then it's going to come back through ATP synthase because we have this lovely gradient that makes the pluses want to equal out on both sides. It'll naturally flow through the ATP synthase, allowing us to make ATP as a result. Right. So all these pluses are going to get moved back in, and these electrons that were moved across through here to drive this whole process, that these are going to collect as well. So these electrons and these H pluses, these can be brought back together with oxygen. We can take half of that, 2 electrons and 2 H pluses, and then these will make H2O. Our final electron acceptor is really important that we get this driven home. Our final electron acceptor in aerobic respiration is oxygen. Oxygen is accepting the electrons in the form of H's recombined back with their electrons again. So what is the final electron acceptor in aerobic respiration, and what product does it form as a result? Well, we've said oxygen is basically what makes it aero in the aerobic. And that we are going to be basically taking half O2, I don't know why that got wiggly all of a sudden, but whatever, with 2 H pluses and 2 electrons, and that all of that's going to allow us to make water as a product. And it's how we're maintaining that gradient of H pluses on the outside, that proton motive force. The whole reason we can maintain the proton motive force is by getting rid of these waste products at the end. Oxygen is absolutely essential in doing that, and it forms water as our waste product. In patients with conditions that impair oxygen utilization, such as chronic obstructive pulmonary disease, so they are not able to do this as well, right? They can't do this as well. Which aspect of cellular respiration will be affected? Our ability to get rid of our protons and our electrons at the end of the electron transport chain will be impaired. So what's that going to affect? Is that going to affect our synthesis of glucose from pyruvate? That's a fake thing. We don't make glucose from pyruvate. We make pyruvate from glucose. That's glycolysis. So this is essentially the opposite of glycolysis. So that's not even a real thing in aerobic respiration. So in cellular respiration or aerobic respiration, the transfer of electrons through the electron transport chain, this is going to get impeded. We can't transfer electrons through our electron transport chain as well because we won't have that gradient to drive everything. Will we have a reduction of oxygen to form carbon dioxide? Oxygen does not form carbon dioxide. That is a separate issue in the Krebs cycle and in that intermediate step prior to the Krebs cycle. You make carbon dioxide from the carbon molecules from the food that you ate, not from the oxygen that you're inhaling. Or the production of ATP in the cytoplasm. We don't make ATP in our cytoplasm. We make ATP on the inner mitochondrial membrane. So B is our correct answer. All right, this is a review of the entirety of aerobic respiration. I've gone over it a few times now, but this is a pretty good little review setup for this. So I do recommend looking over this and making sure that each of these major steps makes sense. There's 4 major steps. There's the 3 biggies, the glycolysis, the Krebs, and the electron transport chain. And this is it's not what I meant for it to do. Oh my gosh, okay. This is, I didn't know that could do that. This is our intermediate step that I keep talking about, where we are basically converting pyruvate into acetyl-CoA. You will make NADH and you will make CO2. But you won't make any ATP or anything here. So that's that step there. So great people create energy. So great would be glycolysis. People is that pyruvate oxidation, pyruvate into acetyl-CoA. C, this is the citric acid cycle, or Krebs. It's the same thing. And then E is the electron transport chain. All deals with energy production. My nose ring is getting crazy. All right. So our questions are, how many ATP molecules are produced directly during glycolysis from 1 molecule of glucose? So you do need to know these. So for glycolysis, you will make 4 ATP. All right, you will use 2 of them. So it's a net of 2, but you will make 4, right? Your intermediate step, you won't make any ATP. Your Krebs, this one will be, I have a brain fart for a second. This is 2 net. 1, I think it's 2 overall for each acetyl-CoA that goes in there. And you'll use 1 of them. So it's 1 net ATP, I believe, for each acetyl-CoA, but you have 2 that's going through from each glucose molecule, remember? So that's where that part gets tricky, and I try not to focus too much on that, but you should probably know the glycolysis one. And the reason I bring up the glycolysis one is specifically because, remember, fermentation is going to rely solely on glycolysis, and we are going to talk a bit about fermentation, especially in the lab portion of the course. That's a really important identifier for a lot of different bacteria, is how they can ferment certain sugars and not others. And so, yeah, so this is 4. If in patients with a condition, patients that have a condition that affects mitochondrial function, which is where aerobic respiration occurs. Right, we're making ATP, such as mitochondrial myopathy. Which stage of aerobic respiration, okay, is the most directly impacted in terms of ATP production? All right, so we made it through these. This is like 2 net. This is like 2 net for the entire glucose molecule, right? For an electron transport chain, your net will be about 34 ATP. So which one makes the most, do you think? Which one's going to be most impacted? Electron transport chain. Glycolysis, you have a net 2. Pyruvate oxidation, you don't make any. Citric acid cycle, you have a net 2. Electron transport chain is going to be the one that makes the most ATP and therefore the most significantly directly impacted. It's also what happens on the actual inner mitochondrial membrane. All right, so the last one, this is the last one for the whole review, is photosynthesis. We have light. This is definitely light. Coming down onto some, oh my God, I keep forgetting that does that. We have light coming down onto a membrane that has, these are pigment molecules this time. And then these pigment molecules get excited. When that happens, you can move our protons back out. And then those guys can come back in and allow you to do your ADP to ATP stuff. This is essentially what the light reactions do in photosynthesis. In the light independent reactions, you're essentially taking this ATP or the dark reaction, call them dark reactions, you know, the Calvin cycle, whatever. You're taking this ATP that you made from the light reactions, and you'll put ATP plus CO2, and you're essentially going to make glucose. That's the gist of photosynthesis in the two parts, the light reactions and the Calvin cycle. All right, so light acts wonders producing oxygen. Light, L, of course, let's refer literally to light, the sun, light energy leading to ATP production, it's acts. Then W is splitting water, which is using the light reaction in order to split molecules. That's going to allow us to generate the H pluses to create that proton gradient. We don't need to worry about that as much for our purposes, but that our whole point is to supply the Calvin cycle, okay? And ATP that you made in the light reactions will literally allow you to drive fixing carbon dioxide into that 6-carbon glucose molecule that we can use for making energy and building parts of the plant, okay? So we have the light reactions. Again, to summarize, we're using sun to make ATP through a process that's very similar to the electron transport chain, but without having to gather the electrons. We're just using photons. And then the dark reactions, which are also called the Calvin cycle, we're using that ATP from the light with carbon dioxide to make glucose. So now let's go answer our questions. What is the primary function of the light reactions in photosynthesis? Does it actually synthesize carbohydrates from carbon dioxide? The light reactions don't do that. That's the Calvin cycle. Is the point to capture light energy and convert it into chemical energy? I would say so. That's the light reactions, right? Do we generate oxygen from carbon dioxide in the light reactions? We do not. That's going to be happening during the Calvin cycle, because part of that is also glucose plus oxygen is a waste product for them. And then D, to break down glucose for energy, that's aerobic respiration or cellular respiration. The whole point is to make glucose so we can put it into our mitochondria, but we have to make the glucose first, and you can't make the glucose without carbon dioxide and the energy that you get from the light reactions, which is ATP. All right, and then our last question, an herbicide is designed that impairs the function of pigment excitation in photosynthesis. Which aspect of photosynthesis could be directly impacted? If you can't get the ATP made from the sun, that's what I'm assuming. So will this affect our Calvin cycle's ability to fix carbon dioxide? No, because this, if you're impacting the pigments, you're impacting this step right here, not the Calvin cycle step. Will it affect production of ATP and NADPH in the light reactions? Yes, absolutely will affect ATP production. The transport of sugars to other parts of the plants. This shouldn't affect that. I mean, you'll need energy to transport the sugars, but if the herbicide is affecting this part here, over here, with the ability to capture sun's energy, that's not what's being affected directly. And then splitting of water to release oxygen, that's not what's happening as from the sun energy on the chlorophyll pigments and things like that. So if you're affecting the ability to excite those pigments, you're affecting the ability of essentially to pump those protons out and create that gradient to make ATP synthase work and make ATP. That's that. Oh, okay, so that's that for our review on chapters 9, 25, and 10, amalgamation. I think we still only have Brandy. Brandy, do you have any questions for me? Okay. Well, thank you, Brandy, for joining me today. And for those of you who are watching this afterwards, I hope that you find this helpful. If you guys have any questions for me when you guys are studying for your exams, please let me know. Otherwise, we have a Wednesday and Thursday this week for our Unit 2 reviews, which is basically not me reviewing, right? But it's you guys reviewing. For my online class, I will have all of those resources from all of your peers posted in a discussion board, and I will send you an announcement about that. I'll actually send that and make that available to all of my sections. And with a disclaimer, of course, that's made by your peers. It's not made by me. And so I haven't curated everything very like specifically, but it should be a pretty good resource. Things like Kahoots or little mini reviews and things like that for you guys to help with studying. So, all right, if you don't have any more questions, I'm gonna call it a day, and happy studying, everybody.